Auzubillahi minash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Assalamu alaikum and good afternoon, everyone. I hope you all are doing well. It's my pleasure to welcome you all in this webinar series called iccbs Talks, which we have initiated as per advice of our very dear teacher, Professor Dr. Mohammad Iqbal Chaudhary, Director iccbs. During this series, iccbs alumni working in top institutions around the world will deliver their lectures, share their research and academic experiences in cutting-edge fields of science and technology. Today is the second webinar of this series, and we have very eminent, prominent alumnus of our center. Professor Iqbal Chaudhary is currently traveling to Kingdom of Saudi Arabia for official visit, and he has sent his very warm greetings to our very competent alumnus, Professor Dr. Hidayat Hussain. To introduce Professor Dr. Hidayat Hussain, I request Director Acting iccbs Professor Dr. Farzana Shahi.
Thank you, Dr. Hina. Assalamu alaikum. On behalf of Director International Center for Chemical and Biological Sciences, I'm very pleased to welcome all participants to this webinar series under the topic iccbs Talk, which is organized by International Center for Chemical and Biological Sciences. I'm pleased to introduce you with our today's speaker, Professor Dr. Hidayat Hussain from Leibniz Institute of Plant Biochemistry, Germany. Professor Dr. Hidayat Hussain received his PhD degree from HEJ Research Institute of Chemistry, International Center for Chemical and Biological Sciences in May 2004. He did his postdoctoral studies from the University of Paderborn, Germany and University of Maine, France during 2004 to 2007. And then he worked as senior scientist at the University of Paderborn, Germany from 2008 to 2010. He also worked at the University of Nizwa, Oman as associate professor during 2011 to 2017 and Humboldt guest professor during 2018 to 2020 at Leibniz Institute of Plant Biochemistry, Germany. Dr. Hidayat was also a visiting professor at Scripps Institute of Oceanography, San Diego, USA for 5 months in 2017. Currently, Professor Dr. Hidayat is working as a group leader and head of NMR section at Leibniz Institute of Plant Biochemistry, Germany. He has 354 publications with impact factor around 1100 along with 10 international patents, 3 books and 12 chapters in books. According to Stanford University database, his name is included in the top 2% of the most cited scientists for 2020, 21 and 22. He is a section editor-in-chief of current issues in Molecular Biology journal, along with member of editorial board of several international journals. His research interests include biology-oriented synthesis of bioactive molecules, biosynthesis of natural products, and biodiversity, along with characterization of natural products produced by microorganisms and plants. Now, I'm very pleased to invite Professor Dr. Hidayat Hussain for his talk on combating cancer natural products and pseudo-natural product-based strategies underpinned by medicinal chemistry. Yes.
Okay. Thank you, Dr. Farzana and Dr. Hina for inviting me for a lecture. And so thanks for Sir Iqbal. And it's my pleasure to give a lecture in my home institute, and we are proud to be HEJian, to have PhD from HEJ. So just let me share my, can you see my slide?
Yes.
Yes.
Okay. Thank you. So today I will just talk, combating cancer. So just because we are working some biosynthesis, also cancer, Alzheimer's disease, also malaria, but today I only focus on nature products and pseudo-nature products based strategy that we, recently, published or under the pipeline of publication. Just before starting, Dr. Hina, can you see the second slide? Because sometimes slide is hanging.
Yes, we can see.
Okay, so just importance of the nature products, plant nature products are the language of life, if you see any process in the universe, you can see some type of chemical reaction involved. As we breathe, we eat, we taste, we walk, so some type of chemical reactions. So also nature products, so any drugs you see, they're mostly on nature product based derived drugs. And you can see here, I mean, from 1981 to 2014, so most of the hop drugs are nature products or nature products derived. And the new terms also introduced by Professor Waldbaum in Germany is called pseudo-nature products. The pseudo-nature products are those compound nature products which cannot be biosynthesized by plants or any natural, I mean, microorganism. So in these pseudo-nature products, you can combine some pharmacophore, active pharmacophore, which is not any biosynthesized, which is called also, I mean, hybrid nature products. So we also did on some type of these compounds also. And the one way to discover drugs is called accidental discovery. And it's true also some biological, and biology, most of the discovery also is called serendipity. But we are, I mean, you see the Dr. Arno mentioned that, the most common discoveries, why not we discover drugs by, accidental mixing, I mean, everything that we can do it. But we are doing, some rational thinking. It's called chemical diversity. So diversity exhibited by library of different chemical entities. So there are so many ways to generate chemical diversity that nature can do it. First is different chemical, I mean, classes of compound are produced by different biosynthetic pathways. I mean, some flavone or xanthone. Also merging of different biosynthetic pathways in one compound. This is called hybrid compounds, just to mix two biosynthetic pathways, or flavone or flavonol. Also functional group modification, you can see sometimes ester aldehyde, combination of monomer, dimer, sometimes you get monomer, dimer, and occurrence of open chain to a ring close compound. So, I mean, there's other way also, but I just have here 4 or 5 ways. And I give you some example then, I mean, how we can learn from nature, and the same in strategy we can use in synthetic chemistry or medicinal chemistry to generate some, active compound. So the first is combination monomers. So we are working on, fungi. So we got these, some naphthol or naphthoferone, or I mean, benzene derivative. And you can see these two monomer like 10, 11, 13. So these monomer we got, and later we got also dimers. So the mixing, like, the mixing of these two compounds through different dimerization. This is how nature teach you, and we can do also the same dimerization in our lab also. Similar also we got, the naphthol derivatives. And also we got ortho-para coupling, ortho-coupling, and para. So all, coupling we got from, fungi. But the meta-coupled we didn't got. Maybe is, hydroxy group is, not a meta-coupled, directing group. So this is just to show you that how we can learn from nature. Similarly, one plants we were working, so we got these two compounds, 2-methyl-anthraquinone and lafacol. If you can see these, and we also give ones just to the prenyl group synchronization. And following this one, we got 2 dimer ones. So the mixing of anthraquinone, one with CC and one with COC. So this, this possibility is possible, to get from, from nature. Similarly, we have, macrolide, tetrahydrohalmidiol, and also this like a fatty acid. And if you see the correlation of these, so this is intradimer of this, halmidiolic acid. So sometimes, if you do careful isolations, you can do, you can isolate both, monomer as well as dimer. So this is, example of, some way of diversity generation from nature. And similarly, you can see the 4 as hydroxy, alkyl chain compound, and 8. And you can see the, correlation between 2. This can be biosynthesized, from this 4A. We didn't get this one, but there's a correlation. So cyclization, is example of open chain to the ring close, compound. Similarly, the same example also 9 to 10, hydroxylation, and then cyclization, and then hydroxylation. So how nature can do the same thing we can do in our lab also. Now, coming to our topic, so what is cancer? I hope everybody's know it. What is cancer? But just to refresh your minds, so the human body is made of trillion of cells that normally grow and divide over the course of our life. When cells are abnormal or get swelled, they usually die. So cancer starts when something goes wrong in the process. So old cells do not die, and instead of growing up control, forming a new abnormal cells. So we have, in our human body, more than 200 different cells, and 200 different cells, can generate 200 different cell of cancer cell line. And the problem, normally, in our human body, around, 50 to 70 billion cell die every day. And a million cell also make up. So, but the problem is when a cell supposed to die, they refuse to die because of the mutation. So these mutation, generate cancer cell lines. So, and also cancer is the second leading cause of the death. So this is just to explain you. And also, nowadays the one treatment is called one size does not fit for all. So, the historical one size does not fit for all cancer treatment approach doesn't work for everyone, and sometimes has done more harm than a good. So instead of using this strategy, we need to use the personalized or targeting treatment. We have to target that proteins which is only present in the cancer cell, not in the healthy cell. So you can, kill or inhibit that, if you inhibit the function of that protein, so automatically cancer cell can be treated. But with the one size fit all, one can, kill also the human, the normal cell or the healthy cells also. So we have to, focus on the personalized or targeted treatments. And just before show you this one, the cell cycles, because in my lecture, mostly, we use, the compound they're targeting these drugs. So mostly drug or the cell cycle based drugs targeting, like, sometimes G2, this phase like this, there's too many, phases or sections or pathways in the cell cycles. One is called like S, G2, M, and G1. So the mostly drugs, targeted these type of cells, because the main purpose of the drug that the replications of the cells or mitosis should be stopped of the cancer cell. This should not replicate into 2 cell or 4 cells. So the main, this is the anti-cancer drug discovery, mechanism. So Taxol or Faclitaxol or, the Vincancalide, Vinclastine, the most targeted are G2M phase. So, and also there are so many enzymes like, cyclo or protein cyclin D, or, it's called Aurora-kinase A and B, or also microtubules, all we'll discuss. But just to show you the cycle, that, this is the, most important, anti-cancer drug discovery targets, that we have to target mitosis to stop the, preparation of the cancer cell line. Now, the first start, so, there's one plant, it's called Frankincense, and also it's called Boswellia, the botanical name. And it's present especially in Arabic countries, only Arabic countries, also in India. And the importance of this plant, people use it, people eat it for treatment of cancer. So we start with this cancer, and this plant is mentioned 22 times in the Bible. And there's one verses also that frankincense, gold, and myrrh, 3 mentioned in verse, were the 3 gifts from the wise men, the magi of the kings, brought from the east to celebrate the birthdays of Jesus Christ. Hazrat Isa salam, to celebrate the birthday of Hazrat salam, so somebody gave a gift, frankincense, gold, and myrrh. So it means the frankincense is plant, myrrh is plant, and gold. So it means gold is expensive. So the importance of the frankincense and myrrh's medicinal importance is equally important, as the gold. So that time, so that's why they give a gift. So you can see the importance of the medicinal value of these plants. And this is a tree. You cut this tree with a knife, and you can, after 15 minutes, you can see, this resin. And similarly, we have also in Pakistan, like we have one plant, kikar. So if you cut it, you can some resin. So this resin have a lot of, medicinal importance. Especially cancer or other. And they have a lot of uses. They have, they call it like magic plants. So they have lots. And the frankincense names derived from the French words pro incense, and it's traditionally China, all everywhere. And the treatment is, fever, asthma, cancer, everywhere. So I cannot read all because of the shortage of time. But to make, we also publish on this, the importance of this, plants recently. And to make a longer story shorter, the main use is to use these plants: cancer, inflammation, diabetes, arthritis, and enhanced memory. And mostly, like in Arabic country, people, especially the pregnant women, eat these type of plants in order to the new baby should have, enhanced memory or intelligent power. And still we are working related to this compound, the plants for cancer, diabetes. But today I will only discuss the cancer. And the most important compound they are isolated from this one is called Boswellic acid. So this looks like ulanilic acid, but they have carboxylic acid and this C4 positions, totally different. And they have a lot of uses. This all patented uses. And the expensive, like they have around 300 euro 5 milligram. So we isolated, this compound, for the plants and proceeded this one. I will not show the, synthesis, this all, we synthesize some pseudo nature products because the original nature products, the original one we isolate is not so active. So we just, make derivation that we learn from nature, like phenazine might be increased. So we tested again pharynx carcinoma, ovarian carcinoma, colon, and malignant myeloma. Against 4 cancer. We synthesize, more than a lot of derivative, but I can show you only the active one. And you can see by introducing the phenazine ring, the activity against, especially this cancer A375, malignant melanomas, increase up to 3.8 micromolar. And similarly, you can see here, by inserting these type of thiourea derivative or nitrogen atom, the activity also decrease, enhanced to 9.3 micromolar. Also, especially we have observed in this triterpene that if you insert nitrogen atom, the activity is enhanced. For example, this one, piperazine, the activity is enhanced up to 2.1 micromolar. So this is just, and also some compound I didn't show here that show activity less than 1 micromolar, but we have in detailed studies. So that is why we are focused, because of the confidentiality issue in our institute, we cannot show those structure here. Similarly, you can see we also as I told you, the nature can do dimerization. We also synthesize some dimerization of these triterpenes. And the monomer was not active, like more than 30 micromolar, and the dimer was active, against, malignant myeloma up to 0.9 macromolar. So the activity enhanced, more than 20 times with dimerization. So the same thing we learned from nature also. And similarly also with this, dimer also activity is 1.3 micromolar. And also this also active again colon cancer with 2.6 micromolar. So this is, and again, I show you this example for a purpose that, the ring is totally clear, so the activity, we lost activity. It means we have to intact all the 5 rings intact and then make a derivative. So if you disturb any ring, so we lost activity. So, this is just to give you a few examples also. Also, we synthesize, some other derivative with the same compound, like with lactam, insertion with lactam, testing with breast cancer as well as prostate cancer. So the activity, 10.8, and also with this one, with the alpha beta saturated, ketone system, 4.1 micromolar also. Also with this, removing the keto group or the hydroxyl group, activity also increased with 3.1 micromolar. So this is, few example, as I mentioned you earlier. So we show example only, only few examples that and the activity, the compound will show activity less than 1 micro, we don't show here at the moment. Now, following the same verses of the Bible, we also selected myrrh. So myrrh is a similar plant, similar, the trees. If you cut, so the you can get the same type of resin, which is like in Pakistan, you call good. So good type resin. So and we isolated, It's called polydipine triterpene. It's also called unusual cyclized triterpene. There's only two rings, so maranol B and B, A and maranol B and maranol A. And the activity is a moderate toxicity, I mean, more than 50 macromolar. So we decided why not to make some dimers. So, I mean, by making dimers, the activities increase up to 40 times. You can see, the activity, like, for example, 7 compounds which have keto group here, as well as the both sides of the keto, as activity enhanced up to 8.9 macromolar. Especially 1.1 macromolar. So the mostly compound here, they show activity less than, 10 macromolar. So, and the original compound like this one, the activity was, less than 100 macromolar. So activity is increased more than. So this is just, we can learn from nature, and sometimes we can get, get compound in a large, small, especially in larger scales. One can try for dermatization, and also one can try, to introduce some nitrogen atom or sulfur atom or some other, ring. So this is just, few example that, we work with chemical diversity that can generate some active compounds in our group. And the third one is mentioned in Bible is gold. So we cannot wear gold, but I wish I could, that person sitting on the gold. Now, coming to the next topic that we, work on is called Aurora-kinase inhibitors as anti-cancer agents. So Aurora-kinase are enzyme and protein. So, these enzymes are involved in the cell cycle, which is, uses replication for the cancer cell line to divide into 1 to 2 and then 4. So error in mitosis can provide a source of genomic instability that is typically associated with tumorigenesis. So many mitotic regulations are aberrantly expressed tumor cells. So, some abnormality in mitosis that generate, cancer cell lines. So these protein, which is Aurora-kinase A and BAC, could therefore make a useful therapeutic targets. As mentioned earlier, then the cycle, there are some enzyme and which is called, mentioned that is, targeted therapy. So we have to target those protein which is mentioned, which is present in only in the cancer cell line, or which mutation take place in the cancer cell line. So these kinases called Aurora A, B, C. So represent a family with such targets, and similar small molecules inhibitors have been shown to block their function. So, and these kinases related to acute myeloid leukemia, this is leukemia cancer cell, type of cancer cells that affect the blood and bone marrow, and also chronic myeloid leukemia, so cancer of the white blood cells. So this is, so that's why we target these enzymes in order to get, Aurora-kinase inhibitors. And also, how these, enzyme, create, cancer. So protein phosphorylation is the most common and important post-translation modification. How the protein phosphorylation takes place. So this is a protein. So you have ATP. So from ATP, one phosphorus, attached to the amino acid of hydroxyl group, and they change to ATP. So this phosphorylation create problems. So protein kinases regulate the biological function of protein via phosphorylation. So these phosphorylation can be two types. Either, good way, for example, it can, inhibit, cancers, or in a bad way, they can also, create cancers also. So mutation and the site of protein phosphorylation, it may lead to occurrence and development of many cancers. So, and also protein kinase regulate majority of cell pathways. So there are so many pathways. And kinases also serine, threonine kinases means it means that these type of kinases, create phosphorylation at the threonine, these type of amino acids. So these are the important, the drug targets. So that's why we want to inhibit these drug targets. And we synthesize some quinones. So I will not show the synthesis of this one, so only the biology. So we have naphtha quinone we have, And so we have orthoquinone, the 110, and also we have one anthraquinone. And this we synthesize, initially we want to synthesize about, for the malaria, but we also tested cancer against different cancer, and we tested against different cell lines, acute myeloid leukemia, small 13, and these 2 cancer cell lines, chronic myeloid leukemia, so 1 cancer, and also breast, 2 type of normal breast, and 2 of triple negative breast cancer, and also colon cancer. And you can see the activity, the compound 1, they have, almost active against every cancer cell line. Except K560 cancer cell line, they are active less than 1 micromolar. Similarly, 2, also, they have methyl groups here. They also are active. Especially these 2 cancer cell line, SCT 1954 and 116 against, less than 1 micromolar, also against, breast cancer 4.1. So, and also 3 which have chlorojuglone. And also this is, is not naturally available. Synthetically modified with the chloro group here. They also show activity on the cancer cell line. Which is called breast cancer, it's 0.99, and also with other like, the leukemia cancer cell 0.18. So you can see the activity, these are, it's all, almost all, compounds were active except, the compound 9 or 8, majority. But, based on this activity, we just selected 2 compounds to proceed for detailed mechanistic action, compound 1 and compound 11, because they are the most active compound. And we proceeded, so just to check either these compounds show Aurora-kinase A inhibitor or not. And for this, there's one called markers, phosphorylation of TAC3. So TAC3 is transforming acidic COL3 is a protein, is a marker. So we have to check is this inhibition, because phosphorylation of this protein can, cause cancer. So that's why we try, and you can see compound 1, 3, and 4, 5 inhibit the phosphorylation. You can see they inhibit phosphorylation. From this study, we understand that they can, this type of compound can inhibit, Aurora-kinase A. And also we tested the same compound, which is active one, a direct enzyme, direct inhibitor. For example, Aurora-kinase NB. So we tested, the activity of this compound directly enzyme inhibition, and you can see the activity is nanomolar. So compound 1.09 with Aurora A, and Aurora B 0.3. And you can see the selectivity. So 3, 4, 5, 4, 10 fold selectivity. So the mostly compound active against Aurora-kinase A. And similar, this is, mentioned the previous slides also. And also we tested like, the 2 compound we tested also, we already mentioned the inhibit phosphorylation also, because we also tested again his histone H3. This is also another substrate of Aurora-kinase A, because we want to make a difference between, Aurora-kinase A and B, which one is active. And from this one, you can see the H3 population, they inhibited only in high concentration. So it means that compound 1 is primarily inhibits Aurora-kinase A than a B. So this make a difference between one. And also we, based on further, mechanistic study, we also tested, it's called NMR based, saturation transfer NMR. So with saturation transfer NMR, you can predict which part of the molecule can bind to the protein targets. And you can see this is the original spectra, and this is the aromatic part. So these aromatic parts has, enhanced. So it means this part is, is directly linked to the target, target protein. And further validation also takes place from the docking study. So both compound, the naphthaquinone and the anthraquinone. So you can see the both hydroxy group, the carbonyl hydroxy group directly, attached to, bind to the alanines 213, and also, the compound 11, which is anthraquinone, is pushed deeper in the activity. Also, OH group makes hydrogen bonding. So this is, further validation that, this type of compound can bind to Aurora-kinase A pocket. And further, study also, we tested these 2 compounds. So, I mean, another mechanism, how these, and you can see here, this is damage, so this is control. So you can see it's called, it's called, I mean, bipolar spindle. So if there's bispindles is also, indicated with this type of, with this type of protein also is 2 here in control. But you can see the treated sample, there's mono spindle, because if you have bispindle, it means the cancer cell can replicate and, change to two daughter cell. But here is monospindle or multispindle. So it means they cannot, go to the replication. So they stop the mitosis, and then cancer automatically can die. So, so that's why it's another target for this compound that, they cannot allow, so they stop the cell cycle and, and the mitosis, and the cancer cell line, automatically die at this stage and cannot go further. So this is another, and also you can see from this study that these two compounds, compound 11, so they, induce abnormal mitosis. So what is abnormal mitosis? Means cell gets stuck in mitosis and they cannot go out, usually kill themselves. And also the the important drugs like, Taxol or vincristine or miblastin also, these type of, they have similar mechanism also. So these, these type of, drug have very good, very good activity. And also we tested, these type of tested further, if they have, how they do for, apoptosis. And you can see here, compound 111 is also, induce apoptosis here, and even in the low concentration also. And also they cleave also PARP. So PARP is a protein, so it means they cleave cancer cells through PARP cleavage also. So this is, that, this is another mechanism for this one. And also they have, this also confirmed from the inacts, stainings, because these type of proteins, so they can combine. So this is further validation of, this type of, cancer cell line that they can kill during mitosis. And also compound 1 induces more cell death. So it means they can kill cancer in the mitosis, but 53.9%, and compound 1 29.6 at high use concentration. So this is further, validated that these type of can kill cell line during the cell cycle and also during mitosis. And also, the further we also did, FL3, so FM-like tyrosine kinase inhibitor. So these also another targets, protein enzyme. So I will not go in detail, but these, these are targets also associated in cancer cell lines, especially these type of genes. So this is gene. So this, this is also gene involved in, the cell cycle. So we prepare some compound in order to inhibit this type of cancer cell line. And also we prepare, around 80 compounds. There's some screening. I will not go in detail. And we just come to the short that we have some chalcones. They have very good activities. And especially, I can see here, all chalcones that we tested here, out of these chalcones, chalcone 4, they have very good activity against, these type of leukemia cancer cell, and MOB13, 0.22, and 0.24. And so the activity against cancer cell line is less than micromolar. And you can see the structure of this one. They have allyl group. So allyl group normally we use in protecting group in organic synthesis. But here this allyl group increase and activity, against this cancer cell, more than 130 times. And for instance, this, this is MV4 ones 160 times, and with THP 1,000-fold. So this, so one should not ignore like the activity of this, especially the allyl protective group. Even the small, functional group can, affect a lot of, activity. And this is not also surprising because, allyl group also present in the garlic, and there's some compound in garlic which have very good, anti-cancer effect. So it's already present. And we tested also further if this, if this inhibit the FL3 signaling, because FL3 is, I mean, important genes in cancer. So we have to inhibit this FL3 is, I mean, the genes in order to get, treatment for the cancer. So this is can be done by ARKs, a different protein, or STAT5 protein. So I mean, you can see these two compounds. I mean, this compound also inhibit the protein, phosphorylation of these two or both. So also possible. And we also did, direct inhibitions in biochemical assay, which show 5.7 macromolar. So I mean, from this is compound that inhibit FL3. And with the docking study also, this confirmed that this docked or this fit to the pocket and ATP binding site are also fit to the FL3 kinase inhibitors. And further also show further study also this compound also that induction mitosis arrest. So it means we tested if this compound can induce the mitotic arrest in the because prolonged mitotic arrest can lead to DNA damage and apoptosis. Mean is a one type of drug mechanism, so the cancer cell cannot grow further. And also this type of compound inhibits cell growth due to cell cycle arrest at G2M phase. So this is all study confirmed that this type of compound can kill cancer during the G2M phase. And also we tested phosphorylation. So this is another marker. I mean, because from this one is you can see the activity of 4 and the facilitaxel. So the activity almost similar. So charcoal 4 inhibit microtubule polymerization because microtubule is I will show the next slides also because this type of compound can microtubule also is another target. For example, a taxol or the vinca alkaloid also inhibit microtubule because microtubule is involved the structure, basic structure and the cells. So, and this type of microtubule is responsible, either they can be replicated in 2 cell or not. So it means inhibit microtubulation means they can kill cancer also. And similarly also you can see they also inhibit, enhance the phospho-H3. This is another marker which further confirm that they can inhibit FL3 genes for cancer therapy. And also, this type of, I mean, I will not go in detail. So, but this type of, especially this type of inducer, a mitotic inducer, and you can see here that either they can, they can inhibit the mitosis or premature exit from mitosis. Both are the mechanism for the anti-drug discovery, and both are good for a compound which can show anti-cancer activity. And here I can show you the microtubule formation. So this is microtubule. So microtubule, which is gave basic shape to the structure. So in DMS, so you can see this type, this type of thread structure is present, thread 1. So this type, because this thread structure is responsible for the any cancer cell line to replicate into 2 cell. But you can see with the treated example, treated cell, this type thread, structure is finished. It means they cannot go, into replication or cell cycle or mitosis. So the cells, the compound, I mean, the cancer cell automatically die after treating this type of compound. So this is microtubule is another target, for the anti-drug discovery. And similar, you can see the further docking study also confirmed that they can, they can bind to the paclitaxel or vinca alkaloid or colchicine binding site. So, but these compounds bind to the colchicine binding site, and aromatic ring of the this chalcone occupy the same region as occupied by the trimethylcellulose. So this is, I mean, this is this is the similar mechanism as the vinca alkaloid or colchicine binding. So they have same mechanism also, and also. Chalcone was able to replace colchicine significantly at 3 different concentrations. So 75%, 80%, and especially the standard was 93%. So they have also, further confirmation that they have bound to the colchicine binding site. And also, the apoptosis, I will not go in detail, that they induce PARP. So it means they kill the cell through PARP cleavage. And also it's confirmed from the, apoptosis for the another, which is called annexin B bonding site. So you can see these cells are going to the G2 different side. It means they can go to the early and death apoptosis. And this is also most important. The chalcone group, because drug resistance is major issue in the cancer drug discovery, and you can see here chalcone 4, which have synthesized, also show activity against MOL13 drug resistance. So the resistant cancer cell line with 0.2 macromolar, and the standard was 1.9. So it means that they have more activity than a standard drug against, especially against the resistance. So it means they can, they can work also the drug resistant approach also. I'll give you one example also, because there's many ways drug resistant, but the two are more common. One is called drug target. Drug target alteration means when the cancer cell line change the target of the target protein structure, so the drug cannot bind to the targets. And the most common is drug reflux. So what is drug reflux means? For example, a drug reflux means when the cancer cell line express pumps in their surface, so the drug cannot enter to there. For example, this is red targets and this is drug. And you can see here, so the drug can easily, in a normal way, they can, bind to there. But if there's express pumps, mean drug resistance, so if you give a drug, so the drug pump up. So these type of pumps, pump up the drug, and they cannot bind. So these type of, and every type of drug, the blue, green, so they go out by pumping because of the drug resistance. So this all, is not possible. But what our drug, our chalcone 4, despite of the drugs resistance, just all pump out. But our drugs I can show you, but our drug can, because even despite of the drug resistance, this drug can directly bind into, the drug targets. So this is, another, best strategy that, that because our chalcone show activity against drug resistant, cancer cell line. So this is one example also. Now, I just have few minutes, so 4-5 minutes. So we also work in triple negative breast. I will not show in detail triple negative breast cancer also. So what is triple negative breast cancer? What is name? So triple negative breast cancer is primarily names, so absence of 3 targets, and these targets are called hormone estrogens, progesterone, so HRT protein. So these targets 3 are missing, so that is why it's called triple negative breast cancers. And also, so that the problem is, with the triple negative, the hormone therapy or other therapy cannot work. So the only therapy is possible that is, called chemotherapy. And also, how I entered this project, I was at Scripps, so we tested some compound in the US, Texas, and around 130 compounds. And around 130 compounds, we have, sorry, I cannot show the structure because still, the lot of compounds now in the, testing case. There's 2 compounds, we call it compound G. The activity against selectively a triple breast cancer, 0.6, 0.2 macromolar. And similar also this compound 0.4, we call it compound H. They have also high activity, especially triple negative breast cancer. There's some activity, especially in our institute also. And we tested now around 200 different, compounds. So, some are, synthesized, some are nature products. And the mechanism they show you, normally we predict is pentose phosphate pathways, but was still under investigation. And around 200 different compounds, so they were synthesized, so they, tested. But the important thing is that now we have 3 compounds, which is now in preclinical trials. So we tested now, and still also we maybe now 5, 6 still will be ready for review. But now on the moment, we have 3 preclinical studies on the, and also, so this is, this is end one. And also there's last topic I cannot show you because there's also another one compound we have called the woman 1. They have very good anti-cancer. This is also in preclinical trial. So I will not show in detail, but coming to the end, so the conclusions that we have, that how does biodiversity add value to human society? So normally we say that plants talks to the environment. So, and also, so we have to understand the talking of the environment. So we understand the talking of the environment, it means that we can understand that which type of plant can be used for what type of diseases. So biodiversity can generate chemical diversity, and chemical diversity can generate, medicinal diversity, and can be laid into new medicine. And the pseudo-natural products, we have to prepare compound which cannot be biosynthesized, and we should learn from nature. So this all, we can learn. So the end, so there's a can in the cancer, means we can beat cancer by scientific innovation. And the end, there's some, these slides show that there's not a one-man show, so that, a lot of people are involved, like, in UK, in US, so there are a lot of people which test in our compounds. And also in the end, I thank to Dr. Hina, Sir Iqbal, and Dr. Farzana for inviting me to give a lecture, and thank you for your attention.
Thank you so much, Professor Dr. Hidayat, for a wonderful, excellent talk. Certainly, you are doing very good job in terms of creating diversity in the pseudo-natural products. So I think now the floor is open for the question. So I can see, yeah.
Hello, Assalamu alaikum.
Yes, Amjad Sahab, you have a question? Yes.
Yeah, this is Dr. Amjad Hussain. Thank you very much. First of all, to all the HGRian, it's wonderful to see the HGRian on the same platform after a very long time. So my salam and very good afternoon to everyone who are listening to my voice. Secondly, I have a question for Dr. Hidayat. Sir, you have mentioned that there are, you have shown a number of compounds that you have synthesized, like the dimer, the hybrids, but of the quinones, your precursor were quinone, but what my experience about quinones, conjugated quinones, that they are, most of the time they are showing activity against the normal cell line. They are toxic. So have you evaluated their potential against the normal cell lines? I mean, for the hybrid or for the dimers? My second question is that the drug that is multi-targeted, that is usually regarded the best one, but at the slide number 52, you mentioned that your compounds, they are inhibiting certain points in the mitotic process. So which particular, have you studied the mechanism, have you conducted the mechanistic pathway, that which particular step in the mitotic process these compounds, they are stopping, are they inhibiting to stop the proliferation of the disease?
Yeah, okay. I mean, thank you for questions. There are two questions. So first is, you mentioned quinones always toxic. It's true. Before, I mean, we call it quinone consider was like an abuse in drug discovery. Nobody work on it because of toxicity. But now, recently I have seen 2019, I have one from in Italy, has one drugs from quinones now is, already approved market now. So, and also we tested quinone in the normal cancers. They are not toxic. And also the same compound now in China are now preclinical trial, so in vivo study. So we will have now research study, but with the normal cancer silent toxicity, they are not toxic. So fortunately. So, I mean, we are lucky we are not toxic. But as I mentioned before, the quinone was abused in drug discovery, but nowadays people are working, they are not toxic as before. So with the new technology. And also as mentioned, mitosis. So some compound we know, we selectively, study the mechanical study of the mitosis, but we only know they have, there's some, but still we, as I mentioned, some compound, we only know they can do induce mitotic arrest or apoptosis, but still detailed studies going on in China for the detailed investigation. We don't know at the moment. with mitotic studies.
Okay. Okay. Thank you, Dr. Saab.
Thank you, Dr. Amjad. Dr. Hidayat, we have a couple of questions in chat box. One is how to reduce the cytotoxicity of synthesized compound in BJ cell line. I think he's asking about the reduction of toxicity in normal cell lines.
Okay, normal cell. I mean, synthesize. I mean, this is journal questions, but definitely you have to work, structure activity relationship, which function group, as I mentioned, especially the allyl group, which is, normally protecting group organic synthesis. So this allyl group also decreased toxicity. I mean, especially, also, especially the toxicity also increased cytotoxicity, around 1,000 times. So sometimes one should not, ignore the normal or the simple, functional group or any, groups in the drug, especially in medicinal chemistry. So, I mean, one should work with structure activity relationship, but from literature, one can learn which function group is important or which is not. But in our case, especially in triterpene, nitrogen atom is most important to enhance anti-cancer activity.
Right. Okay, so there is one more question. Have you discovered any one natural product that is active against different type of cancer and cancer targets?
Sorry, I mean, which have you discovered? And okay, that is active against different type of cancer and cancer targets. As I mentioned, I mean, in this lecture I show you few, but in the triple negative breast cancer, so the two compounds, the three compounds, clinical trials, and these compounds also, not only triple negative breast cancer, but also in other breast cancer. But in quinone, I have also show you that these compound test against leukemia cancer cell line, breast cancer cell line, colon cancer cell line, so and also different type of leukemia cancer. So, but in the especially in the triple negative breast cancer, so we identified compound which is only against active triple negative breast cancer, which is almost good for any drug selectively kill triple negative breast cancer cell line.
Okay, I think we have one more question in the chat box, but I really don't understand this.
What are some of the current research area and use of nature product and pseudo nature product? Yes, is again general question.
Yeah.
So I mean, nature product is used to treat, everything. We cannot go. Normally, I say we cannot go without nature. We have to follow nature, and nature product is everywhere. So we have to follow nature. If you see the example of Corona, so before, nobody want to work on nature product, but when Corona came, everybody now in Europe working on nature product. We have to follow nature to get drugs to treat any disease. And this is also mentioned in Quran में mention है कि हर एक बीमारी का इलाज मौजूद है. And also इसमें भी मौजूद है. जो कुरान मजीद में कुछ पैगंबर जो है प्लांट से बातें करते थे, प्लांट बताते थे, कि वो so I mean the nowadays also just to in Urdu कुरान में मेंशन है कि हर एक कुरान में कि इंसान के हर एक चीज कायनात में खुदा की इबादत करता है. तो अब इबादत जो है वो मौलवी जो है इबादत लेते हैं. साइंटिस्ट मींस दे आर टॉकिंग टू एनवायरनमेंट. सो प्लांट्स टॉकिंग टू एनवायरनमेंट, बातें करते हैं. अगर वो बातें हम समझ जाए जिससे पैगंबर समझे, तो हम इसलिए अब ये बहुत ज़्यादा डिस्कवरी ये हो रही है कि बहुत ज़्यादा वो इंस्ट्रूमेंट आ गए हैं जहाँ से प्लांट्स की बातें आप समझ जाएं और आपको पता चलेगा कौन सा प्लांट किसके लिए है. सो वी हैव टू फॉलो नेचर. विदाउट नेचर वी कैन नॉट गो.
Excellent. Thank you so much, Professor Dr. Hidayat. And with this, I would like to thank all the participants for joining in, and especially Professor Dr. Hidayat Hussain for giving us your precious time and giving us this wonderful lecture. Looking forward to more of your lectures in future.
Yes, thank you again. Sorry, I just mentioned a few things. Thank you so much, Dr. Hina, Dr. Fazana, and Sarik Bal for giving me opportunity, especially for HEG, because what we learned so far is just because of the HEG. I mean, teachers, especially, I still remember the time of the HEG because of the library, because उस टाइम पे सूफी साहब थे, हम इतने बुक पढ़ते थे कि लोग सूफी साहब से नहीं पूछते थे, हमसे पूछते थे कौन सा बुक कहाँ रखा है. So it's because of the HJ library. We are, I mean, because of the HJ teacher and because of the HJ library. So I advise to students to follow HJ library for learning.
Excellent, excellent. I think such a wonderful advice for our new generation. Thank you so much, everyone. You have a good day ahead.
Thank you.
Bye.
Thank you.
Salam.
Thank you. Bye. Thank you.